And our next speaker and the final talk for this session is Mart Last, who's at the MRC Laboratory of Molecular Biology in Cambridge. He's going to be telling us about general pre-trained networks for large-scale in situ studies. Over to you, Matt.
Yeah, I need a couple. Oh. All right. Oh, yes. Okay. Hello. Hi, everyone. My name is Mart Last, and I'm a postdoc with Matteo Allegretti at the MRC-LMB. And I will talk about general pre-trained networks for cellular cryoET. And I mean general pre-trained networks for feature detection in cellular cryoET. And the reason I think these are useful and interesting is in light of things that we were just shown, like automated lamella preparation and very fast tilt series acquisition, the scale of cryoET is increasing quite rapidly. So whereas maybe 10 years ago we were looking at 5 tomograms, we can now look perhaps at 5,000. That introduces a lot of opportunity, but also a bit of a problem, which is that sifting through 5,000 tomograms is not a trivial task. And so I think segmentation is one way to deal with that. But when I talk about segmentation, I find that people often link segmentation up to visualization. And that is one thing that you can do with it. But broadly, I think segmentation can play a very large role in how we deal with growing data sets. So a couple of things that you can do via segmentation are particle picking, where segmentation and particle picking to me are essentially the same thing. It's you go from a tomogram to a coordinate or from a tomogram to a mask. Particle filtering, visualization, identifying particles in their context using biological information offered by feature detection networks, and a couple of other things. And so I want to show examples of these through the lens of, oh, that's going to take a lot of clicking, sorry. Through the lens of a project that I've been working on, which is called Easy Mode, where the idea is to make feature detection and segmentation very straightforward in order to make the biological features in these very complex data sets computationally accessible. And I guess that many of you are familiar with membrane seg, which is a very good example of a way to go about this, in my opinion, where membrane seg is a pre-trained network trained quite broadly, and you can just throw it at the data and it will give you membrane segmentations. So the thinking here was, if we have membrane seg, but for a whole lot of other features, you can then throw all of that at the data and sort of summarize everything that's in the data set and find out what's where. To do this, I've been compiling a large training data collection where we tried to sample tilt series from all over the place. So right now we're up to 4,600-ish tilt series from about 25 different species. A lot of that from Empire, without access to which this wouldn't have been possible. Some from the cryoET data portal, and then also a lot from colleagues around at the LMB. And what that gets us is a very broad collection of tomograms that we can sample very diverse representations of different features from, and then use that to train. And the idea there is that if you train on these 60 different sources, then when you apply the resulting networks to the 61st, the new novel data set, it should also work there. The training data set is a little bit biased towards human species, because about half of it, I think, is eukaryotic, and then slightly under half is sorry, not human species, human cell types. But overall, there's a lot of diverse stuff in there. So what we have at the moment when I made this slide, because by now we have a little bit more, is networks that detect most of the usual suspects in cellular cryoET. So that's ribosomes, microtubules, actin filaments, intermediate filaments, chaperones, Volt complexes, lipid droplets, mitochondria, cytoplasm, nucleus, all that sort of stuff. So most of the big things that you might want to identify in order to figure out what's going on biologically in a particular tomogram. The way we train these networks is we annotate, we train a network in 2D, and then in 3D, we test that network against the whole training collection, and then we visualize everything in 3D to see what works, what doesn't, and then we enter another round of iteration. And after a while, we end up with networks that seem, at least visually, to annotate the target of interest quite accurately. So in this case, we're looking at a tomogram from Patrick Hoffman from the MPI Frankfurt, where we're using the ribosome segmentation network on De discoideum is the species, to annotate what is ribosome and what isn't. The idea then is that you can take the segmentation output, turn it into coordinates, and use that for particle picking, subtomogram averaging, and if the network works, and it does indeed annotate ribosomes fairly specifically, what should come out is a ribosome. That doesn't guarantee that you're not picking other things, but at least when it comes to approximating some ground truth about the particle in cryoET, I think subtomogram averaging is one of the best ways. And so we do that. Then we repeat that for a bunch of other species, always taking that species out of the training collection or using data that was never in the training collection to begin with. And if we can reliably detect ribosomes using this tool, then we consider that network validated and we upload it to our server. I forgot to mention, the reason we call it easy mode is the idea is to give sort of free and simple access to these networks where the weights for these networks live on a server. And if you install the software, you can type easy mode segment ribosome or whatever feature you're interested in. And you don't need to think about where the weights come from or exactly how inference is done. It should just reliably work. So that's ribosomes. That's maybe one of the easier targets. And then we do similar stuff for nuclear pore complexes. Again, in data from Patrick Hoffman, De discoideum. We do the same for actin. And we do the same for a large number of common cellular complexes. So that's the T-ring chaperone complex and Volt complexes. And then we do a similar sort of validation experiment for the networks that segment organelles. So for complexes, you can do subtomogram averaging and get a structure, and that is a way to figure out, is the annotation accurate? For organelles, you can't, because you can't really average mitochondrion, although I've seen André Schwartz try. So we do it differently. We annotate the ground truth. We compare the network outputs to the ground truth, and then we measure the precision, the recall, and the dice score. And this is plotted here for a couple of common organelles: cytoplasm, nucleus, mitochondrial nuclear envelope, cytoplasmic granule, which are these 20, 30, 40 nanometer gray blobs that you see in cultured cells all of the time. Over there, if anyone knows what they are, and I think Tanmay might be able to figure it out with his new technique. Let me know, because I don't actually know what they are, but you see them all over the place. And so we measure these validation metrics on at least 4 different species, again, in order to assess whether the network output is general, or maybe is it overtrained on just yeast or just something. And we find that overall, the overlap with the ground truth labels, which weren't trained on, is fairly decent. So the idea is that if we have this collection of networks, then we can use that to do to support in situ structure determination workflows. So here's an example using data from Kenneth Aces from a paper from this year, where they looked at mitochondrial heat shock proteins that get synthesized in mitochondria that are undergoing folding stress. What they did and what we replicated is use template matching to find these heat shock proteins, HSP60, HSP10 complexes, and then constrain the picks and say we're only allowed to pick particles inside of a mitochondrion and not outside of it. And that should help to get rid of false positives from template matching and end up with a relatively pure set of particles. They did it with manual annotation. So the only thing we're doing here is replacing that manual annotation of mitochondria with automated segmentation. And so just to show some example outputs of easy mode, on the left is a slice from a tomogram. Panel B is a 3D visualization using the pre-trained easy mode networks that weren't trained on this data set. And then in panel C, you see cyan and orange particles overlaid on this 3D scene. And the cyan and orange particles are the hits from template matching. Depending on how well template matching goes and what angular search you use, etc., you can end up with better results than this. But overall, you see, and this often happens, that with template matching, you also pick membranes or ribosomes or ice particles, etc. And so if you have pre-trained networks to say, right, filter this particle set to just the relevant bits of biology, you can then compare the subtomogram averaging results that you get. And if you use all the particles, we ended up with a mostly sort of artifactual bit of membrane density, I believe. Whereas if you filter the picks to this region of biological relevance, you end up with a much better map. Another example is to use segmentation or feature detection to impose priors on the orientation of particles. So back to the DICTI dataset from Patrick Hoffman. Here we segment number of features, microtubules, ribosomes, and again, these cytoplasmic granules in pinkish couple of other ones, and importantly, the nuclear pore complex. And when you average a nuclear pore complex, without a prior on the orientation, they tend to end up in sort of a mixed state where you don't properly differentiate between the cytoplasmic and the nuclear side. But if you have a network to tell you what is the cytoplasm, what is the nucleus, you can then go into the tomogram again and sample these biological features around your particle set. And try to figure out how should my particle be oriented with respect to the things in the environment. So at the top, you see after alignment of the particles, you end up with pretty much a mixed state. So this plot shows you the amount of cytoplasm in front of and the amount of nuclear plasm behind the nuclear pore complex. If they were well aligned, it should be all blue at the top, all yellow at the bottom. So if we take measurements of these organelles around the particles and then adjust the orientation based on that, we can go from a map which was at about 55 angstrom in the top to a map at 36 angstrom, because you now use that biological information to correct the orientation. Final example, this is a tomogram of a purified mitotic spindle in SU verum, a yeast, by Forsen Gao, who is a postdoc with David Barford at the LMB. And here we were interested in the gamma tubulin ring complex, which is the complex where microtubule growth originates at the spindle pole body. And if you want to find a gamma-TOR complex, we can define its location quite straightforwardly in terms of biology. If you just enter the sample, find a microtubule, measure the polarity, now walk towards its minus end, and along your way, you don't exit the sample, you should end up at a gamma-tubulin ring complex position. If you want to find it in some other sort of way, by template matching, for instance, you would need a reference map. You would need to approximately know what the particle looks like. So what we did here was use that strategy based on the biology and on the geometry of the microtubules, walk towards the minus end and then pick particles there, and we find this 24.6 angstrom gamma-tubulin ring complex by simply placing coordinates there. So now to the large scale in situ stuff. This is from the biological project that I'm working on myself. What you're looking at here is U2OS cells with IMPDH, adenosine monophosphate dehydrogenase, labeled with a fluorescent protein. So that's the bundles that you see in yellow, and in red, you've got the nucleus. What's going on here, I think, is complicated. So yeah, my project is about exploring a metabolon that we think might exist. And there are a couple of reasons to think that purine synthesis is orchestrated by a multi-enzyme complex that performs metabolic channeling of reactants between the different enzymes in the pathway. And I think I won't go into detail, but you can ask me later. But what we find is if we inhibit adenosine monophosphate dehydrogenase with a drug called mycophenolic acid, MPA, you see that it goes from free and diffuse in the cytoplasm to forming these very distinct filaments and filament bundles. If we co-stain for other enzymes in the de novo purine synthesis pathway, so PRPS, GMPS, and ATIC, the three ones on the right, it looks like these are also enriched in these filament bundles. And then another enzyme that is known to form filaments by itself and that forms these bundles by itself, CTPS, CTP synthase, can also be detected in these bundles. So what I set out to do during my postdoc was to try to do in situ tomography on these bundles and figure out if these filament bundles have complexity to their composition, or are they just a whole lot of IMPDH all bunched up. So I was already collecting a data set of currently about 1,200 tomograms in HeLa cells, when I also started noticing a lot of IMPDH filaments in data sets from the EasyMo training data collection. So that allowed us to train up a network to detect IMPDH. And then in combination with all the other networks from the model library, we throw all of these networks at the data, 1,200 tomograms, not as big as data sets get, but big enough for it to be very inconvenient to have to go through every single tomogram by eye, which I do actually say we should still do. So this is never a substitute for just looking at the data. But anyway, what we find is these IMPDH filament bundles. So as we zoom through the tomogram again, it's magenta on the right. You see these very distinct sort of discs that form a filament. Each disc is an octamer of IMPDH, which we can now visualize in cell and also map its surrounding within that volume. In this case, we find that there's a lot of intermediate filaments around. It looks like it's fermentin, but we're not entirely sure. Those are displayed in green and then IMPDH alongside in magenta. So sometimes it's just a couple of filaments, 2 or 3. In other cases, you see a big bundle that appears to exclude other complexes like ribosomes or TRIC, or the cytoplasmic granules aren't really in the bundle anymore. And again, there's a lot of fermentin or intermediate filaments around. Overall, I think it's a nice example of if we had all of these pre-trained networks to automatically find the major biological features in terms of which we often describe what's going on in the cell. Are you in the nucleus? Are you in a mitochondrion, etc.? So I think this is a fun example. And it also, segmentation, particle picking, essentially the same thing. Once you turn your tomogram into a segmented map, you can then fill that map with coordinates and use that as the input for subtomogram averaging. So if you're interested in IMPDH and have some in your own data, you can use the pre-trained network for that. We got to 4 angstrom resolution on this octamer. We haven't found other purine synthesis enzymes in these bundles yet, so that remains to be seen. So that's example 4, but it's mislabeled as example 7. And with that, I would like to thank, first of all, Matteo Allegretti, my supervisor at the LMB, and Alistair Burt, who I've brainstormed a lot with during this project. Everyone from my group, support from the LMB scientific computing and EM facilities, and everyone who's uploaded data to Empire, which is a fairly long list of people. This has been super useful to be able to train larger networks on. Then finally, there's a QR code, which if you scan it, it'll take you to a web page where we have the model library in sort of a visual way. So you can click membrane or mitochondrion or TRIC, and you see some example outputs of these networks on different data sets. And that's it. Thank you.